Hello, my name is Amanda Bamer, and I am here to give you a peek into plastic pollution. Cora Becker, my lab partner, and I worked on a project studying the effects of PET microplastic powder leachates on Chlamydomonas reinhardtii algae cultures. Just some brief background on Chlamydomonas reinhardtii. It is a soil-borne terrestrial microorganism. It's classified as a photosynthetic protist, meaning it was exempt from SRC pre-approval, but we were still very careful to follow necessary safety precautions in order to ensure proper handling of this microbe during our project. I do want to draw your attention briefly to figure 1 that shows the growth curve of Chlamydomonas reinhardtii under the conditions of tap growth media, which is what we used in our cultures. So ideally, our experiment would demonstrate the exponential curves shown there. Prior to starting our primary experiment, which we will refer to as experiment 2, we conducted an experiment 1. We wanted to quantify the effects of plastic presence rather than just plastic leachate presence, which we will be doing in our second experiment. So in order to quantify plastic presence, we ended up exposing our cultures directly to plastic powder in the bottom of the tubes to kind of simulate an environment where maybe plastic particles are sitting on the bottom of a puddle and Chlamydomonas reinhardtii are in that puddle. What effect might that have on the local ecosystem? So we did find a significant statistical difference based on the presence of microplastics in the tubes, but we did not find a statistical significant difference based on UV irradiation. We had thought that maybe different levels of UV irradiation, i.e. irradiating for longer, would have the potential to release greater quantities of toxicological compounds, but our first experiment didn't prove that hypothesis. For our second experiment, we thought we wanted to elucidate that a little bit more. So we decided we were going to run our experiment for longer, and we were going to get rid of the plastic powder in the bottom of the tube so that there was nothing confounding our view of essentially data on leachates in particular. So we decided to study whether leachates steeped in a solution of polyethylene tetraphthalate powder irradiated over varying degrees of time, whether those leachates steeped for 2 weeks would have an effect on our algae cultures. I'm going to skip ahead to our data analysis, where you will notice that we didn't find an effect. Because there was no statistical difference and we disproved our hypothesis, we had a couple of ideas on what might have been the case with this. We did think that instrument error, i.e. our homemade spectrophotometer, could have contributed to error in the experiment, as well as contamination of our cultures. We examined our cultures under a microscope at the conclusion of the experiment, and we did find lysed cells as well as bacteria. That's going to create the irregular curve you see at the end of the growth graph on the previous slide. Additionally, we thought that there may not have been a great enough quantity of leachates in the solution. We didn't really have a way to find out whether or not leachates actually steeped into the growth media. We just left the irradiated plastics in the solution for 2 weeks and kind of decided that would have been enough time for leachates to appear. But since there was no way to test for it, that could also point to the fact that there was no difference between our cultures because we don't really know what was actually in the growth media. Because of this, no solid conclusions can be drawn, although I do want to draw your attention to figure 6, where we've pictured similar research done to ours with PVC plastic. The aged PVC plastic did create a significant inhibition on the microbial growth in that particular experiment. So I do think this is something worth exploring further. Additionally, there are environmental implications of our research. I'm going to let you read the slide because it's awfully long, but thank you so much for stopping by.